Welcome to this episode of Life Science Success. For those of you who don't know me, my name is Don, and I currently have a barking dog in the background, just in case you happen to hear that. But just before we hop into the interview with Dr. Russ Lebovitz, I wanted to just play a quick message from our sponsor. This episode of Life Science Success is brought to you by D3 Digital Media Marketing. Dive into the digital world with confidence and creativity on your side. D3 Digital Media Marketing is your all-in-one solution for making a lasting impact online. From the vibrant, engaging world of social media marketing, podcasts, and webinars, to the strategic depths of website development, SEO, pay-per-click, email, and content marketing, we tailor our approach to fit your unique brand story. Let's elevate your presence, connect with your audience, and drive success together. Your digital journey starts here. Visit d3digitalmedia.com to learn more. My guest today is Dr. Russ Lebovitz. He's a seasoned expert in life sciences with over 15 years of experience as a senior scientist and research physician. He's also the CEO of Amprion. And so with that, I wanted to just welcome Dr. Russ Lebovitz. Welcome to the show.
Thanks, Don. I'm so happy to be here. Thanks for having me.
Yeah, it's great to have you on. And, we were talking a little bit beforehand about the overall journey that you've taken. And I always think it's interesting from the standpoint of you have these sort of long timelines where there are not a lot of eyes and ears, that are involved in the company itself. Then you have, the point where, many more people get, interested in everything that you're doing. And given the timeline that Amprion has been around, I would imagine all of those stages have been, exciting as well for you to just kind of see science mature into something that is turning into a business as well. And so would you just mind taking a little bit of time and telling the listeners a little bit about yourself and, sort of what led you to that point of founding Amprion?
Sure. It's a giant zigzag, which isn't necessarily bad. But, my original love and what I pursued was science and medicine. So I trained as a pathologist. And right around the time I was training and getting my PhD, new tools, largely DNA and RNA and some for proteins, were exploding. And so suddenly diagnosis, I trained as a pathologist that really originally was looking at things only under the microscope. And suddenly we had these incredible tools for looking at biomarkers, for soluble biomarkers, tissue biomarkers. And so I became actively involved in the early days of applying DNA and RNA and new protein tools to diagnosis of disease. And that's really where I've always been. I really believe that diagnosis is really critical. You have to understand a disease before you can treat it effectively. So after some time in academia, setting up laboratories, running clinical and research laboratories, I got enticed to the dark side, the startup side, and really got excited that maybe one way to bring some of these new ideas to more and more people would be through companies. And I really love the idea of starting a company. It's not the easiest thing in the world to do, but I've now done it many times. And after, I'm happy to show you each scar from each one, but they're all well worth it. And sometime around 2006, when I was doing this and working with venture funds as an entrepreneur in residence and coming up with ideas for companies, I ran across a gentleman at the University of Texas named Claudio Soto, who had what I thought was an incredibly clever approach to things. And his approach really was for a disease that in the early 2000s we were all worried about. Now no one thinks about it much, but it was mad cow disease. It was a form of a brain disease called Creutzfeldt-Jakob disease, except it could be transferred, it looked like, from an animal to a person by eating meat. And so, what could do this? And so we had learned in the probably 80s and 90s that there was a whole different mechanism of disease called prions, really, for which a number of people, but most notably Dr. Stan Prusner at the University of California at San Francisco, showed that prions didn't contain DNA, didn't contain RNA, but a very abundant protein in the brain could actually form a virus-like particle and cause a disease and spread not only between cells, but to people. So that was where we started. And what Claudio came up with was the idea that these prion-like particles, first of all, they're very rare. They're not in abundance, but that they could be amplified millions of times fold by adapting a DNA technology called polymerase chain reaction. And it's just a cyclic amplification where you start with one copy, after one cycle you have two, then four, then 8, 16, and after 30 to 50 cycles, you're in the millions or billions. And so Claudio came up with this idea and it worked. It became the definitive way to diagnose mad cow disease. And in our conversation, he believed that this would have much wider applications for diseases that, like Alzheimer's and Parkinson's, that in many ways looked like mad cow disease or CJD, but we really didn't know, and there wasn't a firm link yet. But he sold me on the concept, and we formed this company to apply this incredible technology to these diseases, totally in the unknown. We each did it part-time. We got grants for the company from the government. The work was done at the University of Texas initially, eventually moving to Amprion's lab when we were set up. And it just took off. And it turned out to be true that we could detect prion-like particles in Alzheimer's patients, in Parkinson's patients. We could characterize them. And by 2017, we had enough. We had patents filed. We had enough data, and a lot of the rest of the world was taking note. And there were a few other laboratories showing similar results. And we decided to jump off the cliff and form a company. And that's how I look at sort of forming a new company. You jump off a cliff and just believe you're going to land on your feet. And from 2017 to 2024, things have gone very fast in the last 7 years. And we're very proud that we have taken those ideas, turned them into products that are helping people today, both in terms of getting an accurate diagnosis, and we're working with drug companies to use that diagnosis to develop better drugs for these diseases.
Amazing. I do think so. I guess there are two comments based on what you just said. I mean, I do think that a lot of entrepreneurs think that, look, I'm in 2017, I'm going to jump on that cliff and jump off of that cliff and form a company. But I also think that not only am I going to land, but I'm going to land pretty quickly and completely safe, no scars whatsoever. You've mentioned to me as well that you have several scars from the different organizations, but I sort of think it's a great analogy that I think a lot of us go into these things optimistically, and then life sort of happens along the way where you're letting the science formulate its ideas and concepts, but you're also coming along with more questions about what could possibly be done. As you've sort of encountered that journey, I guess what were some of the more significant things that you walked away with, as well, in terms of key learnings between 2017 and now?
Sure. I mean, it's a great question. There are so many. You start a company based on science. So first you have to do research, and science, in my experience, it's iterative. You try different things. Every time something works, you move a step forward. Every time something doesn't work, you either stand still, or if it really doesn't work, you fall backwards, and you just have to get up again and believe in what you're doing. So, we in our research lab, we learned, we iterated, we took a while to get something to work, first of all, that would work every time. And then it needed for us, before we would really consider moving into a product, which is a lot of time and expense in itself, it had to work at scale. So there are a lot of things that's maybe for me one of the major differences from doing science in an academic lab and doing science in a company of any size. But in an academic lab, you show that something's statistically significant, and that's important, and you publish it, and that might be the end, and everyone can see it, and that's great. In a company, if you have a disease that affects 1 million people a year and you can't offer what you've done 1 million times a year at a reasonable price and with great reliability, you haven't succeeded. So a lot of our struggles, our challenges have been not only to make something work in an academic sense, but to make it work in a practical and scalable sense. And I'm very proud, not so much of me. If I have done anything well here, it's to find really great people and help them to make all of this work. It's the Amprion team who's done all of this.
Yeah, I mean, it is an amazing thing to do as well, to formulate something that at the end of the day, helps people, with whatever disease or whatever struggle that they currently have. What sort of made you move, though? I mean, if I look at your background from pathology, to now, what made you, one, want to move in this direction, and then two, what really made you want to focus on neurodegenerative diseases as well, right? What made you want to focus there?
Absolutely. What we didn't discuss earlier is when I was in doing this work in academia with tools really related to DNA and RNA, it really focused on cancer. That was our knowledge of cancer. Cancer is a disease largely of DNA mutations in cells in the body. And you can have many of them, but it was so hard to track them until we had tools. So, I watched molecular approaches to cancer, both diagnostic first and then therapeutic, just take off from, I think if I remember, it was maybe early 90s when the first drug that was designed against something that had been learned through DNA and looking at all of the tools we had, first drug was miraculous. It was against a disease called chronic myeloid leukemia. That had been a giant problem that suddenly could be controlled. And that, for me, lots of bells and whistles went off that we were making progress. And so I watched that field explode from the inside. And maybe 10 years later, it was still exploding when I found the tools for Amprion. But Amprion was just a whole different journey. It was a different mechanism of disease, but for something equally important in the sense that it affected millions of people worldwide and affected millions of new people each year. So it was a chance to, when you're around when something really important happens, it's a great feeling. And even, you're a cog in a very big machine, but it's a great feeling. And to see the beginnings of that in another area equally important, it was just too tempting not to make the jump and say, I trained as a pathologist. It's really about all diseases. I like to joke that, my knowledge is, a mile wide and a micron deep. I don't, as a pathologist, I don't treat people, but the ability to move from one disease path to another was pretty easy. It was all within the training. I knew about Alzheimer's and Parkinson's, but really didn't have any idea of the tools. And suddenly I was sitting face to face with the tools we'd need to do this.
Perfect. Can you tell me about the biomarker asynuclein? And you got to work a little bit on my pronunciation here.
Yeah.
But we just love to know what technologies Amprion has developed to detect it, and the proteins.
Sure. So let's segue into what the major causes of neurodegenerative diseases are and how they progress. So if I said earlier, cancer is a disease of DNA and mutations, to a large extent, Alzheimer's and Parkinson's, just like mad cow disease, are diseases of normal proteins in the brain that have the ability to fold into one or more different shapes. And when they are normally functioning and carrying out what we believe is their intended function, uh, that we'll call that the normal shape. But they have the potential very rarely to form into a different shape. And when they do, under certain circumstances, a number of these misfolded forms of protein come together, form an aggregate, and that aggregate then becomes more stable than the normal. And once that happens, even though it's very rare, it's able to convert all of the normal form of that protein in a cell over many years into the abnormal form. And then when it's full of the abnormal misfolded form, it moves on to the next. And as it moves on to the next, it basically infects a bunch of cells. And after 20 years, you end up with a disease. So what Amprion's technology really was able to do is to look at the misfolding of these proteins to, as I said, amplify it to make it happen in 20 hours rather than 20 years. But it only happens if what we're analyzing has the misfolded form of the protein. So in Alzheimer's disease, there are two misfolded proteins, one called beta amyloid, one called tau, and we started there. For Parkinson's, it was believed initially that the protein is alpha-synuclein. Actually, it still is. A disease called Lewy body dementia is also caused by alpha-synuclein. So there are tools and there are ways to look at amyloid and tau that are already out there. So Amprion decided as a small company to focus on one product where there was a giant need, and that was for alpha-synuclein. We started working on it for Parkinson's, and sure enough, once we had it working, it was pretty amazing that we could take a sample from a person at a very early stage of disease, and it would match exactly and predict what you would find in that person's brain after they went through the disease and died, and you can look at their brains. Because it's really important for neurodegenerative diseases to say that until recently, the only way to know for sure what disease someone has was to look in their brain, and you can only do that after they've passed. And so we wanted to develop tools that would give you that exact same answer while someone was still alive, and even better yet, when it was an early stage of the disease where they had minimal symptoms or sometimes no symptoms. And really the breakthroughs that have happened at Amprion are that we're really able to do that. So for alpha-synuclein, a protein that misfolds to cause Parkinson's, Lewy body dementia, and probably 40% of Alzheimer's patients, we can now detect whether this protein is misfolding and spreading throughout the brain at extraordinarily early stages. Sometimes, as I said, before there are any real symptoms that would allow you to diagnose the disease. So that is a breakthrough. There are other tools we work with in tandem. And so it's a whole ecosystem, but Amprion's part in uniquely addressing the misfolded proteins is a critical part, and we're very proud of what we've done.
Yeah, absolutely. Just interestingly enough, it didn't Robin Williams have Lewy body? Yeah,
Yes.
yeah.
And so these are confusing diseases. So first, they usually end up, with two or three diagnoses, and you figure it out over time. But absolutely, Robin Williams was first believed to have Parkinson's, and then it was understood that he had Lewy body dementia. And this happens a lot. And with these new diagnostic tools, you can tell at the first moment what the disease is, and it helps you predict what's going to happen. In some cases, if there are things that people can do, if it's really early with lifestyle changes, you may be able to slow it down. And if it's early and you have 20 years, but you know you have a disease, there are many clinical trials out there. So you may get 5 shots on goal to actually stop your disease while helping a lot of other people by participating. And soon we're going to have drugs that work from all these clinical trials and work well. So I'm very optimistic.
Very good. So how have your collaborations with organizations like the Alzheimer's Drug Discovery Foundation and Michael J. Fox enhanced the work at Amprion?
Well, I mean, you talk about an ecosystem. So you can't move forward. You have an idea at a little tiny company like Amprion that, barely has enough money to keep the lights on. But if you have a little bit of data and you have a good idea, and you go to someone like the Fox Foundation or the Alzheimer's Drug Discovery Foundation, or there are a number of organizations who believe that this is how progress is made, and you pitch something that basically we can get you an answer within 6 to 12 months that's very meaningful, yes or no. Here's what we would need from you. It's money, it's connections, it's getting the samples. And these organizations are extraordinary, both on the Parkinson side with the Fox Foundation, and on the Alzheimer's side, initially with ADDF, and more recently NIH and the Alzheimer's Association. As you start to make progress, it catches their attention. But you really can't do this without the support of these very knowledgeable advocacy organizations.
And in terms of your progress, the FDA recently issued a rare letter of support encouraging the further use and study of asynede amplification assays. You tell me a little bit more about the letter and its significance?
No, it's very significant. I think that in the last 10 years, for this sort of biomarker, the FDA has put out a total of 25 in the last 10 years. So 2.5 a year. It's not something that happens often. And so it really is the culmination of a lot of work. Again, I'm going to keep talking about ecosystems. So Amprion developed science, then we developed tests where you can measure biomarkers. And the biomarkers that Amprion measures are exactly the misfolded protein biomarkers that you and I talked about earlier, in this case for synuclein. Our job at Amprion is to get awareness of this, and there are several paths. So we can do a bunch of studies ourselves, and that's one way. The problem is, look, we're also a company, so we have a certain conflict of interest that you can never get rid of that. You want things to work. So one of the decisions that I made very early in Amprion's history is that most of our work in validating whether our tests really worked or not were to go to third parties. And that means they knew the patients, they picked the samples, they were blinded to us. Someone sends us 1,000 tubes, they all look the same, but they have a different number. And we read out what we found, and then they tell us whether it made sense based on where they came from. It's very risky. You're betting the farm on your technology. In Amprion's case, it was certainly the right choice, because studies that we did with the Fox Foundation and ADDF and others, turns out what we saw exactly matched what was known about the patients and what was actually in their brains at autopsy. That's great, but it's a very high risk way of doing it, but that's how we have chosen to do our studies. So because of that, there are a lot of organizations that take note, and they believe in it because it's their study as much as ours. And so it really was the Fox Foundation and a group called the Critical Path Institute who looked for new biomarkers. They looked at the data, they went to FDA. We didn't. We stayed out of it. This was really about, if this is an objective biomarker, it just so happens that we largely helped to invent the technology and scaled it so that it could work. So clearly, Amprion knows how to do this. Amprion benefits from it, but it was really all the work of the third parties who went to the FDA. They looked at the objective data together, and the FDA said, yes, this meets a very high threshold. We think we need to see this marker in a number of clinical trials for Parkinson's, Lewy body dementia, Alzheimer's with Lewy bodies, anything where synuclein may play a role. We think that, and they said not only for trials for therapies to make things better, but even trials for prevention for people that we could detect presumably at very early stages before they have disease. So for the FDA to do that, that just says that they've seen enough that they believe that this could be a very useful tool in helping people, and that's their job and our job.
That's a great lead in for my next question, which really is what differentiates Amprion from other companies who are chasing after a similar sort of answer inside of the space. How is it that you're approaching things differently?
Well, we approach, as I told you, we really are, focused on misfolded proteins and the ability in our test to amplify these a million or 10 million fold. Now, why is that important? Well, for a lot of diseases, you have biomarkers either in the brain or in the spinal fluid or in the blood that are important, and they're very, very prevalent. So millions or billions of molecules in a tiny drop of spinal fluid or blood. And that's great. And we have very well established tools that many companies work with. So antibodies are great. They really are the mainstay of diagnosis in fluids. However, that to be able to make an antibody-based detection in a drop of fluid, you may need several billion individual molecules to detect. It sounds like a lot, but given the way biology works, it's not. They're there when they're there. Turns out that for Parkinson's, for Lewy body dementia, for most of the neurodegenerative diseases, in a drop of spinal fluid, for these particles that we are measuring, these seeds, these prion-like particles, there may be 5 to 15 of them in one drop. So way below the technology, way below the threshold needed for established technology. So Amprion steps forward and says, well, we have an amplification technology. We only need 1 or 2, and we can then amplify it in real time to have a million copies, and then we can detect it either with an antibody or a fluorescent dye. But it's the amplification. And because you're amplifying so much, it has to be not only sensitive to pick up 1 or 2, but it has to be incredibly specific, because if most normal people who don't have disease are positive, it's useless. So turns out that that's what Amprion did over all those years when, maybe when we were hidden away in the lab, was developing a technology that was so specific that in the presence of 1 to 10 molecules, we could amplify all the way, to get 10 million or 100 million. And if there were none, you got none. And so why is Amprion unique for diseases where you have a very, very sparse number of these particles? Really, the only technology requires amplification. So antibodies, chemical tests, they just require much more than is there to start with. So it's fortunate that we happen to develop an amplification technology.
Perfect. And in terms of your the current stage that the company is in today, if somebody wanted to go get the test today, is there a test that's available today that can be issued to patients today?
Absolutely. I mean, all of this, look, any test is still only as good as the relationship between the patient and the doctor. In the end, that's the basis for making a diagnosis and a treatment. So while we can detect the molecules, it needs to be done by a doctor with a doctor. And so any doctor in the US can order Amprion's tests. It's very easy. They go to our website, which is Amprion DX for Amprion diagnostics.com, AMPRION DX.com. And once they're there, everything, we have a really excellent customer support group, both for doctors and patients who may have questions. And so, yeah, it is available. And really our next challenge is getting the word out to everyone, and you're helping today, being able to scale to help. There's a million people a year who develop cognitive issues, and probably another 200,000 with movement disorders who notice it for the first time. So 1.2 million in the US alone, a significant number of those that doctors may decide they need this test. And then there's an equal number in Europe, and there's probably 3 times as many in Asia and Africa. So really, our goal is to help as many people as possible worldwide. So we have our work cut out for us, but the unit works, the test works, it gives the right answer. Now we just have to make it available to as many people as need it.
Excellent. And is it run locally, or is it something that runs in a central lab?
It's tricky, to make this work. So for commercial purposes, our regulated test right now is all run out of our clinical lab in San Diego, which has very high capacity. Just, we're already set up to be able to run several 100,000 tests per year. And if we needed to get to 1 million, it's just a question of a few more machines and a few more people. So, it's something that ultimately we would like to be able to make so simple that we can make a kit and that kit could ship out to local laboratories. But it requires a certain expertise that right now we can't make it work. You blink and stick it into a machine. You have to be really careful. You have to run it every day. You have to know your controls, positive and negative. But we can cover what's needed in the US. And eventually, as I said, we'd be happy to be able to have this done locally. But the samples are relatively stable, so they can be sent really from anywhere in the world, not only anywhere in the US.
I feel like it's a journey that most diagnostics take, and some of the most advanced diagnostics that are out there that I can think of, they've all been through this journey, and whether they ever make it to that point of yes, you can run it in the local lab, or it has to be sent back to a central lab that more or less is inside of the organization, I think matters less. But I also think it's important to understand whether you're the physician that's diagnosing or using the diagnostic, or you're the individual that's waiting on the result. It's important to kind of understand the journey that all of that has gone through as well. But I mean, definitely an exciting time to be at, for sure, for with Amprion. So I
Yes.
have a couple questions for you. So what inspires you?
Other people, mostly people who believe in something, that work really hard and use their creativity, people who are persistent, because making something happen to change the world really requires that combination. You have to have a belief and faith system in what you're doing. You have to be incredibly persistent, and you have to use creativity to solve some of the problems along the way. So, I mean, many things, but really it's other people and people doing anything applying those tools. It's not only people who work in science or only people who work in diagnostics, just people being inspired to change the world and make it better.
Absolutely. What concerns you?
That when the world has gotten very complicated, and I worry that, one, a lot of people get discouraged because it's so complicated. Two, that it's very easy to, in a complicated world, one of the biggest challenges is to distinguish truth from falsehood. And look, in some ways it's relative, in some ways it's absolute. But I'm worried that, I work in science. It's actually very simple. There are rules called the scientific method for how you determine if something is true. Other things aren't that simple, but even in science, it's one has to really apply rigorously the scientific method to be sure. And we see all the time, even things in science that people are excited about, and maybe they don't follow all the rules of the scientific method. It gets out, and then people later find that it isn't true, and it tarnishes all of the effort. I mean, we've come so far in the last 1.5 years from painting in caves to doing things that are still painting, but that are very substantial in addition to that. And I don't want that to be lost. And I would like to make sure that we hold ourselves to the highest standard and try very hard to know what's true and what isn't.
Yeah, it takes a lot, especially whenever you have so many pressures put on people to try and succeed, and to continue to hold yourself to a standard where you're going to find the answer, no matter what the answer is. Um, and that answer will be presented, no matter what it is. And that's not an easy thing, I would imagine, in many cases, whenever you have people financing your research, and you got to be responsible to, to many head count that could be inside of your organization as well. Such a
Yeah,
great point.
it's what I explained before, that no matter who I am and how honest I think I am, you know I have a lot riding. I have a lot of people's jobs riding on certain results. So the way I look at it is we have to be objective, and it's the beauty of working in a startup world. Okay, we bet the farm. If the farm floods and fails, we're able to start another one. We did the right thing, and then we get the right to do it again most of the time with something that succeeds. And what we haven't talked about here, and I don't want to underemphasize, is luck, right?
Mm.
20 groups working on things that they believe in equally, and on day one, the science, the clinical hints are equally great. Maybe only one of those 20 work out. And it's who picks the one that ends up working out? That's luck. So persistence is what buys you time to have luck. But I don't ever want to underestimate that, we're all in a little bit of a lottery. We pick something, we work really hard, and if we're honest and it turns out to be true, we'll find that luck. And if not, move on, dust yourself off, try again on the next one.
Very good. Last question. What excites you?
Oh, the future, mainly. Just I see so many great things in the ideas coming through. What people who are younger than me are how they're viewing the world. Um, I'm not a pessimist. I'm very much of an optimist, and I see in my own children, I see in other people's children, I see a lot of very creative, hardworking, generous, and thoughtful people who will be running the world in the future. And I hope they do a better job than we have, but we've tried.
Yeah, absolutely. Well, thank you, Dr. Russ Lebovitz, for being on the Life Science Success podcast. I greatly appreciated hearing about Amprion. And I would definitely encourage people to go out to Amprion's website, so Amprion DX, and, definitely, investigate their products, and, look at the journey that they've been through. So thanks so much for being on.
Thanks, Don. It's been great for me.
Thank you.